Welcome to the NIH Director's Wednesday afternoon lecture series. As I am Dr. Ofelia Olivero from the National Cancer Institute, and I have the honor of hosting Dr. Ken Ramos today, as well as introducing him for his lecture. Dr. Ramos was born in Ely, Nevada, and received his Bachelor of Science degree in Pharmaceutical Sciences and Chemistry from the University of Puerto Rico Medical Sciences Campus. He received his PhD in Biochemical Pharmacology from the University of Texas at Austin and his MD degree at the University of Kentucky. Dr. Ramos currently holds several positions. He is the Associate Vice President for Precision Health Sciences, Interim Dean of the University of Arizona College of Medicine, Phoenix, Executive Director, Center for Applied Genetics and Genomic Medicine, Professor of Medicine, University of Arizona Health Sciences, and he does not sleep, right? Dr. Ramos has received international recognition in the fields of applied genetics and genomic medicine, precision medicine, environmental health sciences, and toxicology. He is an elected member of the National Academy of Medicine, as well as an associate of the National Academy of Sciences. Today, he will talk to us about repetitive DNA sequences in health and disease, gift wrappings for precision medicine. And I hope you all can join us for a reception in the NIH library immediately after his talk. Please join me in welcoming Dr. Ramos to the stage.
Thank you, Ophelia, for your introduction, and certainly it's a pleasure for me to be here to share with you some of the things that occupy some of my time. I did get tired listening to all the things that presumably I do, and hopefully I do well. But as all of you know, we always find time to do the things that carry us with passion, and certainly science and medicine does that for me. The story that I'm about to tell you today is one that began in the early 90s for me, when we made the observation that repetitive sequences, which were sequences that we believed to be dormant in the genome, could actually be reawakened in the context of stress. Those findings, of course, were very much in keeping with the findings of Barbara McClintock when she had reported a decade or 2 decades before that on the stressful response of maize. And of course, as probably many of you know, the redefinition of what we think in terms of genetics and the stability of the genome. Over the course, I think, of our studies, what became apparent, and hopefully something that you will appreciate from some of the highlights and examples that I will give you, is how important these sequences will turn out to be in the context of precision medicine, which is sort of the buzzword that we now use these days to talk about the intersection of molecular genetics, applied genetics, and delivery of patient care. And so I will try to place those things into context and give you some of the examples of how we've tackled that particular component. In choosing the title, I elected to call repetitive sequences, in particular lines, which is really what I work on, long interspersed nuclear element sequences of that particular family, gift wrappings for precision medicine. And the reason that I call that, use that description, is because surprisingly, perhaps disappointingly, when we think of precision medicine today, we really think of biology that only covers roughly 2% of our genome. And despite all of the advances that we have realized as a function of that, that finding reminds us that there's 98% of the genome that we need to begin to try to uncover to even begin to scratch a surface on how we're going to apply what we do in the laboratory in the context of patient care. And so the intent is to give you some examples that basically speak to that. A couple of disclosures before we move forward. We actually have filed a patent for a test and treat paradigm building on some of the findings which will be captured here. And we have been working to establish private partnerships to actually advance novel therapeutics in the space of this particular set of problems. This slide basically captures something that's probably close and dear to your heart because it's essentially the huge accomplishment that we made as a scientific body in completing the Human Genome Project and all the work that's actually ensued, following completion of that exercise. This truly has been one of the greatest feasts in exploratory biology, and certainly, I think, has given us an opportunity to develop the blueprint, the book, essentially, that has guided much of what we do in the context of applied genetics and its intersection with the field of medicine. From that intersection, we basically have been able to realize essentially three major buckets of activity that I think guide what we do. We've applied genome-based evaluations to diagnostic precision, increased diagnostic precision. Some of the best examples I think that we can cite in that particular context relate to precision oncology, where standards of care are now being defined on the basis of genomic insight. You could think of breast cancer perhaps as a poster child of precision oncology, in which, irrespective of pretty much where in the country you are, some form of molecular diagnostics will be utilized routinely to carry out decisions and triaging and management of patients afflicted with that condition. Second important application, I think, of the intersection of genomic science and medicine is in the context of pharmacogenomics. Certainly a science that's not very new. I recall, 40 years ago, taking a course in pharmacogenetics. But the level of insight with which we covered, pharmacogenetics in those days, of course, has totally, completely redefined itself over the course of the last 40 years. So now we're beginning to realize targeted therapies that not only can be used for improved efficacy in managing specific conditions, but perhaps more importantly, in decreasing toxicity and minimization of adverse outcomes for the patients that get treated with targeted therapies. And certainly, I think an area that's going to provide a lot of opportunity for continued expansion as both the public and the private sectors continue to work together to be able to advance that particular space. And then last but not least, the idea that optimized clinical management of patients will require stratification and molecular diagnostics, because one of the biggest problems that we have faced in the field of medicine, and we certainly encounter it as we engage in clinical trials, is the fact that one size doesn't fit all, right? Sort of the mantra of precision medicine, the idea that each one of us is unique in our own way, and that we need to be able to capture that level of resolution in order to provide optimized ways in managing specific, conditions. And of course, a shift away from remediation of problems in the context of health to a paradigm that focuses on prevention. And so one of, I think, the more sombering aspects of this whole story is the fact, as I indicated in my introductory remarks to you, that a lot of what we do, a lot of what we think we understand, really constitutes 2% of what I'm showing here in this slide as 55% of the genome. And that the rest of that genome is constituted by sequences that we understand poorly, though we've made great strides in the last 10 years, and for which understanding of regulatory biology is very, very limited. And so the focus of my talk today is going to be strictly on 17% of that 45% that we understand very poorly and that we call repetitive DNA within our genomes, and for that matter, the genomes of pretty much all species. Turns out that 17% is, or 19, depending on the reference that you use, or 20 even now with some of the redefinitions that have come along, constitutes a set of sequences that we called long interspersed nuclear elements 1. And the majority of those sequences are sequences that have been silenced during the course of evolution, primarily through truncation of the sequences that regulate the activity of these sequences. And as of today, up to maybe 100 of these sequences remain active in the human genome. Turns out that of those sequences that do remain active, because they have retained structural integrity to carry out transcriptional activation as well as pasting back, reinsertion back into the genome, are the sequences that I believe we're going to be able to capitalize on in trying to advance the goals of precision medicine. So these are the gift wrappings that I was alluding to in my title. Notice that the remainder of those sequences are sequences that we call mobile repetitive DNA sequences. That means that under the right context, under the right conditions, these sequences can actually mobilize. But what makes the 17%, the line sequences group within this larger 45% so unique is the fact that lines are the only ones that can do so autonomously. And so it's that autonomy of mobilization that makes them sort of the, I guess, the premier repetitive sequence of the genome in the context of, mobile repetitive sequences. And so the story that I will share with you is one that's taken roughly about 20 years in evolution. And I will begin giving you snippets of our early translational studies that kind of set the stage for clinical validation, both in retrospective as well as prospective studies, with the idea that hopefully we can take advantage of our understanding of that biology to treat and manage disease conditions in which these sequences appear to be important. And so back in the 90s, we reported, made the observation that when you study the whole genome expression of the transcriptome, if you would, of cells undergoing transdifferentiation, a process that essentially guides many of the pathologies that we think of today, including cancer, including fibrosis, including chronic obstructive pulmonary disease, including arthritis, including atherosclerosis, including dementia. There's a lot of genetic reprogramming in those pathological conditions, and when you try to gain insight into the genetic reprogramming events that drive transdifferentiation of cells, line 1 sequences appear to play a very huge role. Back in the day, when we completed these experiments and we cloned and subcloned these sequences, what we discovered was that more than 50% of the sequences that appeared to be upregulated in the context of that transdifferentiation response, whether it's epithelium going to mesenchyme or mesenchyme going to epithelium, appears to be line 1 sequences. And so when we made that discovery, the perhaps naive assumption that we made was that these sequences must actually be either secondary players in the transdifferentiation response or potentially drivers of that response. For those of you who may not be in this field, here is a quick schematic giving you the major mammalian transposable elements in humans. Suffice it to say that the sequences that we are primarily interested in are the ones of the non-long terminal repeat family, of which L1 is the major family member. Remember that this is to be differentiated from long terminal repeat sequences, the vast majority of which, up to 98% of which, are really inactive in the human genome and only remain active in rodents and other species. Also important to the biology of lines is that the activity of ALU elements, which probably are of interest to some of you in the audience, is actually heavily regulated through the machinery encoded for line 1 elements. And so when we made the observation that transdifferentiation was directly coupled to the upregulation of these sequences, we, of course, knew very little about these sequences. And in fact, in those days, when you did a literature search on it, unfortunately not PubMed back in those days. These are the days of Index Medicus, where you sort of plow through pages and pages. What you found is that there was really hardly anything in the literature. If you did that same search today, taking advantage of an electronic search engine, you would find thousands of publications per year in this particular topic. But when you put all the small pieces of information that were available together, what you could sort of come up with is the idea that these repetitive sequences were almost a hybrid of repetitive inert sequences within the genome with the codification machinery that we have grown to be so familiar with in terms of, codification for proteins. And that lying elements actually carried within them a 5' untranslated region that functioned as a regulatory region and had two open reading frames. One open reading frame encoding for what we now understand is a ribonucleoprotein that's very critical to the assembly of ribonucleoprotein particles that get shuttled back into the nucleus for reverse transcription. And ORF2, which is an open reading frame adjacent to open reading frame 1, which encodes for enzymatic activity of reverse transcriptase as well as endonuclease activity, which is used for the nicking of DNA and for priming of reverse transcription, and a 3' UTR with a poly A tail. So in many ways, an architecture that reminds you of conventional gene codification structure. And what's really interesting is that it's only through the course of evolution where reverse truncation of these sequences only allows for copying in reverse mode of the sequences that are inert and that when pasted and inserted in truncated fashion in the genome become what we used to call the junk DNA that does not encode for anything and that for the most part we did not understand its biology. As I alluded to already, about 100 of these sequences remain full length and presumably active in the human genome. And important, I think, and relevant to the remainder of the talk is the fact that many of these sequences are highly polymorphic in humans. And so the assumption that we guided many of the experiments that we designed was that if these sequences are highly polymorphic in humans and if they're upregulated or deregulated during the course of transdifferentiation, they might actually serve to explain some of the differential response that we see. I did not want to be incomplete. I did want to introduce you to perhaps the newest set of discoveries made by the Denil lab with a new open reading frame that was discovered in primates. It's important because it appears to influence the mobility of line 1. It actually encodes in the antisense orientation, and that's going to be really critical because the antisense codification of these sequences has actually been regarded as one of the silencing mechanisms that's utilized by the L1 machinery to regulate rates of transcription in cells. I think one of the comments that was made by a colleague here in the audience early on when I was chatting with him before coming to the podium is the idea, and we're lucky that these sequences, are not so active and that we are, alive and well. And of course, the answer to that is yes, nature is wise. And nature has engineered many multiple ways in which these sequences are basically kept in check. And are not allowed to do the havoc that they could do in the genome. However, when the sequences are reawakened, if you would, they're reawakened through disruption of epigenetic silencing that involves both histone code as well as DNA machinery, DNA methylation machinery to basically maintain the DNA of the L1 sequences silenced. Injury, stress, transdifferentiation forces these sequences to reawake, and when they reawake, a number of things can happen. The conventional understanding of L1 biology is that these sequences, if allowed by the contextual framework of the cell involved in the insult, would actually reinsert back into the genome in what's believed to be random insertion throughout the genome. And if that insertion goes into an inert region of the genome, it would probably be a silent, a mutational insertion. If it goes into a region of relevance and functionality of the genome, of course, then disease could ensue. And in fact, since the early 80s, you find examples in the literature, occasional examples of diseases that have been implicated with aberrant, if you would, line 1 mobilization throughout the genome, hemophilia being the first one that was documented by the Kazazian lab in the 80s. More recently, we have actually published work that implicates a non-insertional biology, and some of what I have to say for the remainder of the talk will be important for that context, so I will reserve those comments for that. We spent a number of years actually trying to characterize the silencing machinery that governs the regulatory expression of line 1 elements, since that became critical to our understanding of transdifferentiation machineries in cells. And so suffice it to say, we certainly could spend a lot of time talking about the contents of this slide. This is one of those slides that, you know, in one frame you capture 15 years worth of effort. But suffice it to say that the major discovery made by the group was that retinoblastoma protein and the machinery orchestrated by retinoblastoma appears to be a major master regulator of the silencing activity of line 1 retroelements in mammalian cells, and that in the context of that transdifferentiation response that I alluded to early on, what happens is that the ability of retinoblastoma to remain in complex interaction with the macromolecular silencing machinery becomes disrupted through changes in DNA methyltransferase activities, as well as changes in histone methylations and histone acetylations in ways that allow for that chromatin to open up and that allow for transcriptional activation of the retroelement, which in turn encodes for the proteins that I showed you in the previous slide. There's formation of those ribonucleoprotein particles in cis, preferential, L1 encoded proteins forming this particle that then gets shuttled into the nucleus for reverse transcription and reinsertion. And that if you want to re-silence the element, you have to regain RB regulatory control in the cell. As you would imagine, based on this sort of very quick synopsis of the complexity of line 1 silencing, what you can surmise is that anything that disrupts cell cycle regulatory control, anything that modulates the activity of the transcription factors interacting with retinoblastoma, anything that modulates the epigenetic machinery in ways that interact directly with that silencing macromolecular complex is going to be a potential impact to the expression of line 1 sequences. And here is an example of the importance of retinoblastoma in that context. So this is an experiment in which we took clones of cells in which all members of the retinoblastoma family had been silenced, and we basically challenged these cells in culture with a chemical carcinogen known to reactivate line 1 expression. And as you could see, there's marked upregulation of line 1 expression in the context of RB loss. You could reproduce this experiment, through native mutations of RB. You could reproduce this experiment by modulation of the phosphorylation activity of RB. You could, reproduce this experiment by modulation of p53 activity. Anything that you will ultimately compromise RB interaction with that DNA sequence will certainly be contributory to the upregulation of line 1 sequences. And then you wonder about what the biological impact is of that. But at the time that we completed much of this work, trying to figure out, this sort of level of complexity at the level of regulatory control for transcription of this element, one of the things that kept, occupying our mind is that we understood little about the reprogramming biology that was involved in transdifferentiation. And so one of the things that we did was take advantage of computational biology approaches, utilizing very simple binary models for predictive biology, Boolean sort of idealization strategies, and we defined what we believe to be a relevant architecture of a line 1 regulatory network. The intent of these experiments was essentially the idea that if we understood proteins and genes that interacted with line in the context of transdifferentiation, we might be able to begin to reverse engineer, if you would, the regulation of differentiation programming in ways that we could then take advantage for disease. Suffice it to say, in the interest of time, that we validated the computationally derived network that I've shown you here in the slide. These are some of the critical players. I'm not going to have time to go into sort of the unique interactions there. Certainly happy to talk to you about specific examples, but for now, I think the message for you, take-home message, is that this regulatory network appeared to be honed in to 3 specific domains of biological activity. And those domains include inflammation and inflammatory biology, a lot of interactions with cytokines, and a lot of interaction with things that regulate cytokines. Extracellular matrix genes and sort of the extracellular matrix machinery of cells, which, as you know, is a very important regulatory signaling pathway for control of differentiation behavior in cells, and cellular metabolism. And so when you kind of put together those 3 domains, you start thinking of diseases in which this might be important. Cancer immediately, of course, surfaces to the top. And in my case, given my clinical interest and background in pulmonology, I started thinking of inflammatory conditions of the lung in which perhaps line 1 might be playing an important regulatory role. And so the remainder of my talk will highlight for you, walk you through our own journey of translational validation, prospective and retrospective validation of line 1 activity in the context of chronic obstructive pulmonary disease and its potential to contribute to lung cancers of the lung. And so why the lung as a potential target? Well, it turns out that the lung is a portal of entry for many agents in the environment, and that the injury associated with the environmental impacts in lung epithelial cells, as well as some of the other cell types present in the lung, involved inflammatory signaling pathways, because a lot of the conditions in the lung really are dictated and defined by uncontrolled inflammation. In response to that inflammatory response, there is something that occurs heavily, and that is epithelial to mesenchymal transition, EMT, a term that's very familiar to people in the field of cancer, very familiar to people in the field of arthritis, because it turns out that epithelial to mesenchymal transition recapitulates a lot of the transdifferentiation programming that I alluded to when I presented the slide that we had studied 20 years before. And so it turns out that in response to uncontrolled inflammation in the lung, there is epithelial to mesenchymal transition, and that epithelial to mesenchymal transition has been directly implicated to either chronic obstructive pulmonary disease or non-small cell lung carcinomas of the lung, which of course constitute the majority of cancers of the lung, about 85% of them. And importantly to the work that I'm going to show you, the idea that this EMT response is actually directly coupled to dysregulation of transforming growth factor beta signaling, both through canonical pathways as well as a non-canonical one. Turns out that there's extensive upregulation of line 1 sequences in metastatic lung cancer, and that many diseases of the lung exhibit considerable heterogeneity, which goes back to the point that I made before about the highly polymorphic nature of these sequences and whether that polymorphic behavior might actually contribute to disease. And so here's the results of an experiment in which what we did was we forced expressed line 1 into bronchial epithelial cells. These are cells that come from the larger bronchi, probably to third degree level within the bronchial tree, and cells that probably will contribute primarily to the occurrence of either contractile activity in the context of COPD, inflammation in the context of COPD, or central tumors in the context of non-small cell lung carcinomas. So this forced expression was actually associated with the induction of epithelial to mesenchymal transition, in keeping with the idea that the reprogramming associated with transdifferentiation might actually be directly coupled to the overexpression of these sequences. There is contribution of both insertional biology, insertional mutagenesis biology for some targets, as well as non-insertional biology to this. Suffice it to say, in the interest of this talk, that the majority of the actions of lines to modulate epithelial to mesenchymal programming in epithelial cells is unrelated to insertional mechanism, and all of the activity is actually seen in the absence of integration back into the genome. And so we started asking ourselves questions about the relevance of this biology to the pathogenesis of COPD. Clinically speaking, this is a very important question to ask because chronic obstructive pulmonary disease has become the third major killer in the U.S. as well as the remainder of the world, in many ways connected to lifestyle factors, environmental exposure, smoking. But interestingly, even today, with the advanced medicine that we have, all therapies for chronic obstructive pulmonary disease are palliative therapies. We basically are treating symptoms. We're not directing these therapies to understanding mechanistic biology nor to the pathogenesis of disease. And so we are now trying to utilize our understanding of the critical role of line in regulating the reprogramming behavior of cells in the context of health to disease transition for bronchial epithelial cells as potential targets for therapeutic intervention in the context of COPD. I think it's important because a lot of the biology that you see in patients with COPD is highly consistent with the biology that you see during altered line 1 signaling in cells. And so I'll give you a couple of examples of an assay that we've developed and some of the results of retrospective experimental study. So one of the challenges in studying line 1 sequences is, of course, the signal-to-noise ratio. You have multiple sequences in the genome. You need to specify zip codes, if you would, address locations to be able to know which lines you're really working with. And importantly, cause and effect, like with many other genetic relationships, becomes really critical. So we wanted to develop a simple blood test assay that would be suggestive or representative of what the activity of lines in the genome were. And so we developed an ELISA assay that basically is established on the basis of ORF1P activity. We have characterized this ORF1P ELISA assay in the context of a biomarker development pathway, walking all the way from its discovery as a candidate biomarker all the way to human population testing, and I'll show you some of the tidbits of results. Happy to answer some questions related to that. So what we did was, in this particular set of studies, we took a very large cohort of chronic COPD patients, COPD defined on the basis of forced expiratory volumes in the lung, which is a conventional clinical definition that you utilize to identify these types of patients. And we stratify these patients on the basis of severity of COPD, and that's where the L1 quartiles data that you see on the slide alludes to. And as you could see is that as you correlate blood levels of the ORF1P protein encoded by line 1 with severity of chronic obstructive pulmonary disease, these levels actually go up. If you were to this is about data from about 600 subjects, and we'll continue to expand upon that because we've become interested in determining not only the correlation between levels of the protein and severity of disease, but the longitudinal trajectory of these levels relative to progression into malignancy for the lung. And I'll speak to that in a few minutes. That particular set of questions become really critical to us given the high level of genetic admixture that we have in U.S. populations. It becomes really critical because the genetic admixture that characterizes the American society is something that for the most part has been largely ignored in most genetic studies. We basically have very broad categorizations of ethnicity. Many of those categorizations are probably incorrect. because they fail to take into account genetic heterogeneity in these populations. And this is going to be highly critical to line 1 biology because of the polymorphic nature, and I'll show you some results that will speak directly to that. And so the idea is that we would characterize the heterogeneity of the U.S. population, in this case chronic COPD patients, to try to understand how therapeutic interventions directed at silencing or sort of control of line 1 might actually regulate trajectory of disease in these patients, if in fact L1 is driving the trajectory of disease, which we believe it is. Line 1 can be used then as a means of stratifying the population, and if you stratify the population on the basis of line 1, and line 1 is playing a contributory role to trajectory of disease, and you intervene with that dysregulation, you should be able to make a difference in this disease. And that's sort of the conceptual framework that's guiding a lot of the work that we're doing right now. I actually believe that in the space of precision medicine, stratification of populations is probably one of the greatest contributions that can be made. And so in the context of lines, I think this is going to be really important because even though we've got 100 full-length sequences, even though we have a huge sea of signal-to-noise ratio in the genome, only about 9 of those lines appear to be active to the level that can actually dysregulate biology and maybe contribute to disease. And so what appears like an insurmountable task of navigating through 600,000 plus sequences becomes manageable when you only have to worry about 10 of them. And so turns out that we're highly polymorphic for many of those sequences. The location of those sequences within the genome is here, and these are the sequences that we are actually now exploring in the context of chronic obstructive pulmonary disease, with a particular interest in genes and loci residing in chromosome 6, which is an important chromosome in the context of lung pathology. And so the results of an experiment in which we compared 4 chronic obstructive pulmonary disease patients to control are shown here in this slide for each one of those loci that I showed you in the previous slide. Suffice it to say that turns out that these cohort of COPDers was polymorphic for these sequences, and that those present in chromosome 6 appear to play an important role in the regulation of severity of disease. And so this is an area where we're continuing to make huge strides. So if you remember, I alluded to the fact that patients with COPD are at higher risk, up to maybe a 3-fold higher risk of progressing into malignancy of the lung. But we all know that there's a whole bunch of smokers that never get lung cancer. And so what that directly speaks to is, of course, the heterogeneity of the population. And the studies that we are doing right now are really being focused on trying to unravel, if you would, the potential contribution of lying, given its ability to regulate the biology of transdifferentiation in the lung, given its ability to induce genomic instability, given its ability to cause havoc in the genome, to maybe drive the transition of patients into chronic obstructive pulmonary disease and then complete that journey into malignancy of the lung. And of course, the idea is that if you can understand the complexity of that biology, you can actually capitalize on that to develop pharmaceutical agents that might actually interfere with that progression. The idea would be that you would treat a COPDer with medications that would prevent uncontrolled transdifferentiation to progress, and that if you prevent uncontrolled transdifferentiation to progress in combination with silencing or resilencing of line 1 sequences, you might actually change trajectory of disease. And so an example that I elected to present to you is an intervention in which we tested the ability of TGF-beta-1 receptor 1 inhibitors, drugs which are already clinically used in the market for the treatment of pulmonary fibrosis, to actually manage line 1 mRNA levels and to correlate the inhibition of line 1 activity caused by these agents with their ability to prevent transdifferentiation and their ability to improve clinical outcomes in patients. And we actually have a clinical trial going on right now, beginning to explore the usefulness of this approach. We're utilizing multiple ways to tackle line 1, either upstream through regulation of TGF-beta, because we've actually demonstrated in our studies that TGF-beta lies upstream of line 1 in the regulation of EMT biology. And we're also tackling the line 1 directly by way of therapeutic modification of line 1 sequences so that you can prevent its ability to reverse transcribe. And so the success of those experiments, I think, is still preliminary, but I think we're encouraged enough by the results of those studies that we think that this is going to be an area where new small molecules are going to be developed in ways that will allow us to directly tackle line 1 and to be more precise in the way in which we're making interventions that guide clinical decision-making for management of those conditions. And so some of you might ask yourselves, well, why is this sort of important? And the answer to that lies perhaps in the newest repertoire given to pulmonologists and to oncologists in the screening for lung cancer. Not many lung cancers are screened for, except with perhaps the notorious difference for breast cancer, but most other cancers, screening is actually discouraged, and lung cancer is another exception. But lung cancer screening has actually one huge Achilles heel to it. And that huge Achilles heel is that the screening is confined to the right age bracket, patients, age 55 to 80, who have a 30-pack up smoking history or who have only quit within the past 15 years, i.e. at higher risk of malignancy. And it's a low-dose CT screen that actually identifies nodules in the lung. And it turns out that the incidence of solitary nodule, identification and as part of the screening presents a huge clinical problem for the pulmonologist and probably even the oncologist, because 95% of those solitary nodules that are identified are actually false positives. And so this sort of invokes all of the complexity that we have to deal with in terms of, are we doing ourselves a disservice by screening patients in ways that are going to basically mostly provide false positive results, increase costs, to our healthcare system, increase patient, dissatisfaction, increase psychological discomfort, and end up in a clinical trajectory that probably is not going to be conducive to increased progress. And so one of the things that we are doing is we're combining line 1 measurements with screening of low-dose CT to see if the predictive power of this screening exercise actually improves as a function of that. And so that takes us, I think, full circle into the whole space of precision pulmonology with the 3 areas that I alluded to in one of my introductory slides. That is the importance of taking advantage of genome-wide evaluations for diagnostic precision. In this case, screening of patients for polymorphic variants of line 1 that might be more predictive of line 1, heightened line 1 activity in that genome. You could do that certainly taking advantage of sequencing, but you could do it taking a proxy, using a biomarker that can give you a readout of that activity. Development of targeted therapies that will increase efficacy because it's going to be targeted directly at the pathology that you're dealing with and hopefully decrease toxicity. This is going to be certainly true in the management of lung cancer malignancies, which are metastatic, in which line 1 appears to be playing a very huge role. It turns out that the majority of what line 1 does is actually directed towards regulation of migratory and malignancy phenotypes. And then last but not least, stratification of populations in the context of repetitive sequences, which is an area that we have largely ignored, mainly because it only reflects the lack of understanding that we've had up until technology cooperated to be able to guide us through new journeys of discovery. And with that, I'd like to close it because I've been told that 45 minutes is the right mark and acknowledge, of course, the fact that one's achievements are never our own achievements. They are an extension of those that we surround ourselves with. So I'd like to acknowledge my family at home as well as in the laboratory, my collaborators, funding agencies, and of course all of you for your attention. I look forward to engaging in dialogue and talking about what I've discussed with you today. Thank you very much.
We have time for some questions. Please use the microphones.
Congratulations for covering an interesting area of chronic obstructive lung disease. So you mentioned there are sequence variants, and there are about 100 you have already identified are using it. Could ELISA technique find those variants, or you have to do the sequencing to look for those variants that might affect the lung cancer in some of these patients?
That's a great question. The ELISA assay is only going to be a crude readout of increased activity or low activity for line 1. If you want to know variants itself, you're still going to have to do it by sequencing.
And also, you mentioned that there are some gender variation. Is there some protective effect for a woman?
Actually, the counter to that, the sex difference for line 1 is really interesting. And it probably has some degree of connection with, X chromosome silencing and sort of the biology of cyst and the connection to lines. Long story short, it turns out to be that males overexpress line 1 more readily than females. So females are better adept at maintaining line 1 sequences silenced. But women that are either polymorphic for high expression variants or that get exposed to things that deregulate the stability of line 1 sequences, once it's destabilized, the response of women is actually heightened compared to males. This is really interesting in the context of COPD because the same biology is reminiscent of COPD, where you mostly see male COPD patients, probably because they're the ones that began smoking earlier. But when you see females presenting with COPD, the severity of disease appears to be enhanced. So that's pretty interesting there. The other aspect, I think, to genetics of line 1 variation that is very critical is that Native American ancestry appears to be protective of line 1 reactivation. And so we're beginning to explore sort of the connection between genetic ancestry and regulation of these sequences. We don't know if the difference between Native Americans and other ethnic groups is genetic or epigenetic, and so we're beginning to sort of plow a little bit in that direction. But your observation is quite on target.
Thank you.
Yeah. So a couple of questions about mechanisms. So the first is, so if you're now transcriptionally activating these elements, are you also potentially activating recombination mechanisms?
By all means. In fact, when one of the consequences of line 1 reactivation is the fact that you are increasing rates of recombination. For lines, that becomes sort of a three-edged sword, if you would, because it's not only lines, but it's going to be the ALUs, the SVAs, and that you need to worry about. Lucky for us, I think, what happens is that there's about 12 mechanisms that would halt recombination in the context of lines that you actually have to have every star align in the sky, if you would, for the full recombination cycle to be completed. But indeed, it's one of the major devastating consequences of it.
So there's increasing evidence that repetitive elements can actually form transcriptional fusions with adjacent protein coding exons. So is that likely to be a mechanism that's at play here as well? So if you transcriptionally activate a repressed element, it now activates downstream exons of a oncogene or something else?
Absolutely, indeed a possibility. We're beginning to actually look into that interactome complex that you've alluded to. That's a great question.
Thank you all for being here, and please join us for a reception by the library. Thank you.